Today we'll be talking about section 12.2a, where we outline how glucose transporters in the plasma membrane of mammalian cells imports glucose after a meal and then exports glucose to provide fuel for the rest of the tissues in the body. But before we can start, there are a few definitions we should get out the way. Beginning with transporters. Transporters ship small organic molecules or inorganic ions from one side of the membrane to the other by changing shape. Next, we should talk about passive transport. Passive transport happens when molecules spontaneously flow downhill from a region of high concentration to a region of low concentration. Now that we got these two definitions out the way, we can start talking about what a glucose transporter is, especially since this is a prime example of a transporter that mediates passive transport. More specifically, a glucose transporter is a protein of polypeptide chains which allows it to adapt several conformations, making it the perfect transporter. One of the conformations exposes binding sites for glucose to the exterior of the cell, while another conformation exposes sites to the cell interior. The transporter then switches irreversibly and randomly between those two conformations. Glucose transporters in mammalian cells are so important because glucose is such a vital source of energy. GLUT and SGLT are both examples of glucose transporters that are found in most mammalian cells.
Here, the transporter is shown in three different conformational stages. First is the outward open state. This is where the binding site is exposed to the outside of the transporter. The ossoluted state, the one in the middle, is where the binding sites are not accessible on either side of the bilayer. And the inward open state is where it's only accessible to the inside of the bilayer. After we eat a meal, we feel energized. The glucose transporters in the plasma membrane help export glucose and provide fuel for the other tissues in the body. After a meal, there is plenty of glucose outside of the cell, so it's easy for sugar to bind to the transporter's binding sites. The transporter is highly selective and will only bind to D-glucose, no others. If the protein decides to spontaneously switch conformation, it will carry the sugar into its glucose binding site, and then it will release it into the cytosol, where there is a lower glucose concentration. If there's a higher concentration on the outside of the bilayer, the glucose will bind more often to the transporter in the outward open conformation, which I discussed previously, so the net transport of glucose remains down its concentration gradient. The net flow of the glucose moves inward if the glucose is binding to the transporter's externally displayed sites, outward if the opposite is true. When we are hungry, the opposite is true. Our blood glucose levels are low, so the hormone glucagon stimulates liver cells to produce large amount of glucose by breaking down glycogen. This newly produced glucose is used to bind to the binding sites of the transporter, and when the protein spontaneously changes in the opposite direction, the glucose is transported out of the cells and is ready to be used elsewhere.